Hello and welcome back. ANP2 Chapter 27 is about fluid issues. We've done so many organ systems and we've talked about so many metabolic processes that were in force and chemical reactions that were in place. It's time to take a step back and think about the overall chemistry with respect to water. And the issues here are fluids, the dissolved substances that we call electrolytes, and acid base. First, let's start with the water. 99% of the fluid outside the cells, the extracellular fluid, is water. The extracellular fluid is basically like a pond in a bag. It surrounds and establishes the chemical conditions of solute concentration for all the cells in the body. There are different fluids in different compartments. The main connection there is the blood and the blood plasma. It is the functional transport system of the body. But that fluid outside the cells, not alive, but maintaining the life within each cell. Water is an essential element of the cytosol as well, that intracellular fluid which contains all the living reactions that keep the cells alive. They all rely on water. That polar molecule that establishes that condition we call hydrophilic, that dissolves and moves, transports all the gases, nutrients, waste products, and other characteristic molecules of the body. Fluid balance is the daily balance between what we take in and what we put back out through perspiration, through urine, through saliva, and other substances. We're going to regulate the relationship between that extracellular fluid and intracellular fluid and balance it with our daily intake and loss of water. The digestive system is the primary source of water gains. There is a small amount, which I'll call metabolic water, that's generated from respiratory activity. You remember, breaking down glucose produced CO2 and water. It is a small amount of our daily water balance. The urinary system is the primary route of water loss, although it is not the only means by which we lose water. Let's take a look at the whole system. Electrolytes include all the ions which are released from the dissolution of inorganic compounds. It's called an electrolyte because a solution with sufficient ions can conduct electricity. Electrolyte balance is where the gains from our diet are equal to the losses from our water excretion. In general, we're going to be looking at the balance between the digestive tract and the kidneys and sweat glands in order to consider the balance of water and fluid dynamics. Acid base is looking primarily at that hydrogen ion, very, very active, and activator for our chemistry, and maintained in the blood plasma at a very, very narrow range. How is it we're going to take the learnings from previous organ systems and add them all up to a homeostatic control of our pH? Roughly, water accounts for 60% of male body weight and 50% of female body weight. That's just water molecules. Most of this is in the intracellular fluid. Water exchange between the ICF and ECF occurs across the plasma membrane. Now, the force that moves water is called osmosis. The process that moves substances combines diffusion, if it moves freely across the membrane, and carrier-mediated transport if a protein channel or if a protein channel plus energy is needed. The extracellular fluid is divided, I think, fairly conveniently into two main bodies. The interstitial fluid of the peripheral tissue. So that means throughout the body, everywhere in the body, there are the cells, and then there's the fluid between them. Those are called the interstitial fluids. The other major kind of extracellular fluid is the circulating plasma, which is water-based. It basically propels and carries blood cells and appropriate concentrations of electrolytes around the body. Now, the extracellular fluid also shows some minor compartments that have their own specialized water-based fluid. Lymph, perilymph, and endolymph. Cerebrospinal fluid of the central nervous system, which we have already examined. Synovial fluid, that special fluid that's in each of our freely movable joints. Serous fluids are those fluids inside between the outer and inner membrane of a serous membrane. Pleural around the lungs, pericardial around the heart, and peritoneal around the digestive tract. Aqueous humor is a special water-based fluid that's found specifically in the eye. The exchange among these subdivisions is across the capillary wall. So water is delivered to regions by the cardiovascular system, especially the vessels, and it is the capillaries that are porous. So this is where water balance is achieved. From the interstitial spaces to the plasma, it can also move through lymph vessels that basically return that more water-based fluid, return it to the venous system. When we look at what's carried around in that water, the solid components that are dissolved are shown on the left. Proteins are not only the most diverse, they are the most abundant, followed with a severe reduction by lipids, those hydrophobic materials that constitute not just our fat deposits, but every membrane in the body. Minerals are those things that give rise to our electrolytes, and you'll notice carbohydrates kind of are a reduced but important component, still measurable. The kilogram amounts are shown in this comparative graph. Miscellaneous includes all the other chemical components of the body, and you'll notice that nucleic acids of the big four are not listed here. The total weight of the nucleic acids, even though they're present in every cell, is relatively small. Over on the right side, you see intracellular fluid, about 20-some liters inside the cells, and the extracellular fluid divided into the majority comparable amounts of these fluids with males containing more by body weight. The extracellular fluid and intracellular fluid, their solute content, let's look at this list. Types and amounts vary regionally around the body and depending on the organ and organ system. There will be electrolytes. Those are the materials that are charged, establish and hold the cell potential, account for electrical differences that stimulate sensitive membranes like muscles or nerves. If a cell can react, it typically is due to the presence of the right concentration of electrolytes and their functioning correctly in movement across the membranes. Proteins are the most diverse and the most abundant by weight. Nutrients are pulsed in by feeding episodes and must be constant in order to maintain metabolism within the cells. And finally, that metabolism generates wastes, so we have to account for them. We call these fluid compartments because they basically behave as very distinct entities. The living cytoplasm of the cell is very different than the fluid that surrounds its membrane and bathes it and maintains its chemical relationship. The plasma membrane, the semi-permeable membrane, and diffusion or active transport are maintaining that very precise balance. Cations and anions, or cations are and anions are plus and minus ions. In the extracellular fluid, we find differential concentrations of sodium, chloride, and bicarbonate. The high concentrations of these three are found outside the cell. The sodium-potassium pump itself accounts for the sodium outside the cell, since it is constantly taking ATP and moving sodium out. Because of that, we expect potassium to be more concentrated inside in the ICF, and there it is, leading the way. Potassium, magnesium, and phosphate are found in higher concentrations inside the cytoplasm. And especially the negatively charged proteins are more abundant and more diverse inside the plasma membrane. The proteins are there constructing and producing enzymatic conversions and forming subunits. They are a part of the living cell. So let's take a look at these comparisons. Here's a picture for intracellular fluid, the cytosol. And so what do we see? In cations, lots of potassium, quite a bit of magnesium, a little bit, that green stripe is calcium. Notice the sodium is a minor constituent. On the anion side, the negative ions are led by proteins and phosphates, with bicarbonate, chloride, and sulfate present in trace amounts. Now, keep that in your mind a little bit and compare that to the plasma of the blood and extracellular fluid. Look at that sodium and chloride concentration. That salt you eat is adding pure ion to your body. It dissociates in water and basically is maintained in high concentrations outside the cell. Notice also that the cations, the positively charged ions outside the cell in the plasma, have trace amounts of potassium, calcium, and other things. I think that bottom bar is magnesium. Over on the anion side, chloride is by far the major one. A few proteins are circulating, and organic acids, hypophosphate, and bicarbonate are also found in the plasma. The interstitial fluid is very high in sodium. Think back to that sodium-potassium pump and its work. The interstitial fluid is full of chloride, as you would expect, outside the cell. Those two dominant ions then leave little room for the trace amounts of other cations and anions, but they're shown here. Now, we talk about fluid compartments. Fluid compartments are really exist in a very, very different way. If you think about it, with the selectively permeable membrane around every cell, here's a little pocket of intracellular fluid, and right next to it is another complete membrane with a little pocket of intracellular fluid, and so on. That means that the intracellular fluid as a compartment is made up of literally trillions of cytosol elements for the trillions of cells in the body, and they are separated by their plasma membrane. So to get from the intracellular fluid of one cell to the intracellular fluid of another, you have to have a transport mechanism to get over there. We have to consider that when we think about how do you regulate the chemistry body-wide. Enter the extracellular fluid, which basically surrounds the outside of these plasma membranes and is essentially continuous throughout the body. So you go around this cell in the extracellular fluid, then you go around this cell in the extracellular fluid, and then this one, you don't run into membranes. The extracellular fluid is the outside. That's why I say it's kind of a very, very specialized pond, and these cells are behaving just like cells in the pond water in relation to the water outside. Osmosis is going to be important, the maintenance of cell size and shape and structure. If we have equal osmotic concentrations, we call that isotonic, and cells maintain a consistent shape and water balance. It is osmosis that's going to adjust minor differences in concentrations across the membrane, because that difference in concentration will cause water to move toward the higher concentration of salt or solute. Basic concepts in the regulation are shown here. The physical principles of the universe can never be escaped. And in fact, what you must do is find a way to operate the cell obeying all of those laws. So, number one, all homeostatic mechanisms that monitor and adjust body fluid composition respond to changes in the extracellular fluid, that continuous bath that's around all the cells, and not to the trillions of separate micropools that are the intracellular fluid. No receptors in the body monitor fluid or electrolyte balance. There is no such receptor. It's a combination of chemoreceptors and osmoreceptors, pressure receptors that add up to our fluid balance. Number three, and this is an important one, a cell cannot transport water molecules by active transport. When we think of the sodium-potassium pump, that's something the cell can do. It can burn ATP, move sodium out and potassium in. That's a capability of that carrier, but there is no such carrier and no such active transport mechanism for water molecules. We're going to regulate the water or electrolyte content, and it will rise if our dietary gains exceed our losses. It will fall if the losses exceed gains. So when we think about homeostasis or health, that's where our water intake and electrolyte intake is balanced with our losses. When we think of unbalanced intake and loss, so for example, the disease diarrhea is characterized by extraordinary voiding of liquids, and liquid is lost, carrying with it a very, very high concentration of electrolytes. And that's what happens when you have an extended case of diarrhea or vomiting. You lose so much fluid and so much electrolyte, the electrolyte gets out of balance, and it leads to a general feeling of malaise and illness. Often a hospital stay and intravenous injection of fluids, normal saline or supplemented saline is necessary to first establish that electrolyte balance before you can go into the healing process. The primary regulatory hormones that are shown here, in fact, all three of them have been mentioned in prior lectures. Antidiuretic hormone is ADH. It functions by regulating the water retention versus water release in the kidneys, ADH. Aldosterone is part of the renin cascade, and it has a water retention effect as well. Natriuretic peptides. Here is one that responds to increased blood volume and increased blood pressure, causing the release of water. ADH, water conservation in the kidney. So this is where you've been working, but not replacing your fluids. You're getting lower and lower in your state of hydration and approaching that line we call dehydration. ADH's effect is to cause the kidneys to retain sodium and, as a result, by osmosis, remove water from the urine and concentrate the water, the urine that is released. This is water retention. Osmoreceptors in the hypothalamus are what detect changes in that osmotic pressure and cause the secretion of ADH. The hypothalamic neurons stimulate release from the posterior lobe of the pituitary. This is one of the places we find that special capillary called the fenestrated capillary, the one that allows absorption and transport of ADH with higher speed than a normal continuous capillary. Aldosterone is in response to the renin cascade, and it responds to rising potassium or falling sodium in the blood. Either one of those can activate the renin-angiotensinogen system, and it basically the rate of sodium absorption and potassium lost along the distal tubule is changed. As you can imagine, if its effect is going to be to retain water, it's going to move more sodium and potassium into the surrounding material, and the water follows the salt. High aldosterone in the plasma causes kidneys to conserve salt, and so water is retained in the tissues surrounding the tubules, meaning that higher and higher concentrations of urine are produced. Natriuretic peptides, just to review, they cause the opposite effects. They reduce thirst. They block the release of ADH and of aldosterone, and they cause diuresis. Now, diuresis means the release of high volumes of water that will lower blood pressure and volume. Here's a kind of a representative illustration of a total water balance, thinking about it on a one-cell basis as a representative of the entire body. That plasma membrane, that circular membrane that is shown in red here, is surrounding the intracellular fluid right here. So this is representative of the intracellular fluid compartments. They exist by the trillion in a human body. And the relationship between this water and the extracellular fluid, the surrounding bathing, the bath that surrounds all the cells, is what we're talking about. Now, it turns out something happens in here that's important on a daily basis. When we think about glucose plus oxygen forming CO2 and water and ATP, something we've seen so many times, that water actually represents a substantial contribution. 300 mils is actually generated. This is not water that we take in our digestive tract and absorb through the intestines. This is water that is a product of our ATP production. So let's call that 300 mils of input that can come to the extracellular fluids. Up here is our 2.2 liters of daily average water intake. So how do we lose water? We lose water constantly from our skin and lungs. The gas that comes out of our lungs is loaded with humidity, and insensible perspiration is that inescapable loss of water from our skin. This is water that's lost from our feces, a very small amount in a person whose digestive tract is working well. Down here is a major loss to urine, about 1.2 liters a day under normal conditions of inactivity. Now, the dotted blue line is sort of a variable. This is the amount of exercise that you engage in every day. If you engage in lots of exercise or lots of hard physical work, if you are working in hot conditions, then of course you sweat a great deal. This becomes a very big arrow under those conditions, and that means that this water intake must increase significantly in order to balance it. So these are the major factors that affect our water balance. This is the way those compartments work. At the top, the balance is isotonic, and the levels show, and the spacing of the dots show, equal solute concentrations. Notice that that's true, even though it's not equal volumes. It is the concentration of the solute that produces the osmotic pressure. So what happens over here if you lose a portion of that fluid? So you decrease its volume. Notice how these dots have gotten closer together. That means the solute concentration in the blue box has gone up. That will cause water, which is more concentrated over here and now less concentrated over here, to move from the ICF into the ECF. That will cause the lowered volume of the ECF to increase. At the same time, the volume of the ICF decreases from that movement. A simple osmotic relationship. Electrolyte balance. This is something that is critical to our welfare and our sense of good health. We have already talked about vomiting and diarrhea, those things that take water and electrolytes indiscriminately out of our body. And we basically have to balance the loss and gain of these electrolytes. They directly affect the water balance because of osmosis, and that total concentration is the concern there. But in fact, individual electrolytes have different jobs. Here are the major ones. We're going to go through this list to see if we can recognize major and kind of cohesive roles for each one. We have noticed that there's quite a bit of variation in the range that we call homeostasis for each one of these. Calcium is an interesting example because over 99% of the calcium in our body is mineralized in solid bone, and that mineral we call hydroxyapatite. The soluble portion of calcium is one that's very low concentration, 8.5 milligrams per deciliter, a very small amount. But its range is small, 8.5 to 11. Its range is only, what is that, 8.5, 9.5, 10.5, 2.5 milligrams per deciliter is the allowable range. Go too high and the body starts making bone crystals. Go toward that 8.5 and the body starts chewing up bone. So each of these has its own important role. Rules of electrolyte balance. Most are caused because of sodium ion. Because sodium ion is the positively charged ion and is present in a basically unlimited amount because we eat salt. When the sodium ion goes, it affects electrolytes all over the body, primarily through osmotic changes. The problems with potassium balance are less common, but they're more dangerous because potassium is essential inside the cell. If the improper amount of potassium is there, then the cells themselves actually are compromised. Not a general effect like osmosis, but rather a specific effect that affects metabolism within the cell. Let's look at sodium. It's called natrium in Greek. That's why we call it, we name it Na on the periodic table. Table salt, the rock we eat, is our source, and we can get pure sodium and pure chloride simply by adding this crystal to our food. It's the dominant cation outside the cell. The sodium-potassium pump, its effects in cell potentials. You remember so many muscle and nerve actions where the critical thing when the electrical impulse arrived was that gates popped open and sodium rushed across. So it must be outside of every cell and abundant. 90% of the osmotic pressure is produced just by sodium. The forms that are common in our body, sodium chloride, because this is what we take in our diet. Also sodium bicarbonate, a form where the bicarbonate ion and sodium combine. Homeostasis. The normal sodium concentration in the ECF is disturbed in this diagram by increasing sodium levels in the ECF. When that's detected, osmotic pressure changes. Osmoreceptors in the hypothalamus detect that, and they secrete ADH to restrict water loss. They don't want the concentration of sodium to go higher. It also stimulates thirst and promotes water consumption. So add some fluid, add some solvent, and the sodium concentration is diluted down. Because of the change in the osmolarity, there's that word right there, the water shifts out of the intracellular fluid, increasing the volume of the extracellular fluid, which decreases the sodium concentration. That decrease returns sodium to its normal range. The opposite occurs when sodium decreases. Osmoreceptors, again in the hypothalamus, will basically stir the release of ADH, and that concentration change, ADH secretion decreases, so thirst is suppressed, water loss by the kidneys increase, and as water is removed from the body, the sodium concentration goes up. In chemistry, we're sort of used to thinking about putting solute in fluid, but always changing the solute amount. Remember that water balance affects those homeostatic ranges just as profoundly as they are the second part of solute concentration. So the body reacts to hyponatremia. That would be what? Low sodium. Body water content rises over hydration from the retention of water, and the extracellular fluid concentration is over less than 136 milliequivalents. I'm sorry, that's hyponatremia. Hypernatremia is over 145 milliequivalents. This is where the body water content declines, dehydration. Plasma volume affects all our concentrations, and a reasonable range of volume for blood plasma is the first factor in blood pressure concerns. If the venous return increases, it stimulates atrial natriuretic peptide and brain natriuretic peptide, having the opposite effects, reducing thirst, blocking secretion of ADH and aldosterone. That also then causes salt and water loss at the kidneys, and we dump some water. That brings the ECF volume down, and with it, the blood pressure. Potassium is an interesting one because here we are, a critical electrolyte, an ion that's mostly found within the cell. Most of it, 98% of the potassium's in the cytosol itself, primarily as a result of the sodium-potassium pump and a critical part of cell potentials. Cells expend ATP to recover the potassium ions diffused from the cytoplasm and return it to the extracellular fluid. The rate of gain across the digestive epithelium, we have to get potassium from our diet, but we do lose some in our urine, and those are the two principal factors in balancing this ion. Hypokalemia and hyperkalemia are shown here. Low potassium will increase the volume of urine that is released, and an endocrine disorder called aldosteronism is characterized by excessive aldosterone, results in hypokalemia. Hyperkalemia, low blood pH, will produce it and interferes with potassium excretion in the kidneys. Several drugs promote diuresis by blocking sodium reabsorption in the kidneys. Here's one of my more interesting ions, because so little is present in the body that is not crystallized in bone. It's the most abundant mineral in our body only because we're vertebrates. A typical individual has 1 to 2 kilograms up to 4.5 pounds, but most of it is in the skeleton. Soluble calcium is a low level. Look at that, 8.5 to 11 milligrams per deciliter, but what does it affect? It turns out it is a cofactor that we have seen in a number of processes. Calcium must be present to contract a muscle. Calcium, remember the dumping of calcium from the reticulum, the sarcoplasmic reticulum, for a muscle to unlock the actin-myosin junction. Calcium rushing across the membrane was necessary when a nerve impulse was affecting the fusion of vesicles with neurotransmitter substance and dumping that into the synapse. And so we're looking here at an element, very, very low concentration, but without it, you can't contract a muscle and you can't fire a nerve. Turns out you need calcium for clotting blood, any number of other things. Magnesium, we find this in bone, doesn't have very much in the body, and most of it is in the skeleton. However, it is a cofactor in the intracellular fluid that affects something as important as phosphorylation of glucose at the beginning of glycolysis, the use of ATP by contracting muscle fibers, and on and on. Magnesium is effectively reclaimed by the proximal convoluted tubule, and the daily requirement is relatively low. It's easy to get enough magnesium. Phosphate ions we've seen before, ATP, the phosphate that's on the nucleotide. Phosphate is required for bone mineralization. Basically, 3/4 of a kilogram is bound in the mineral salts of the skeleton. And daily urinary losses are very low, 0.8 to 1.5 grams. Inside the cell, it's required for these high energy compounds, the activation of enzymes and the synthesis of this one, nucleic acid. So it's critical for our genes. In the plasma, it's reabsorbed from tubular fluid along the proximal convoluted tubule as a constant reclamation. Its concentration in plasma is up to 3 milliequivalents. So it is a small trace a presence, but very important to our overall health. We've talked so much about positive ions. What about the negative? Well, the dominant one's going to have to be chloride. Every time we have a sodium ion in our diet, we have a chloride from the salt we eat. It's the most abundant anion and balances the movement of ions that are negative. So if we're moving if we're moving bicarbonate, a negative ion, we often exchange it for chloride. We saw that in the transport of bicarbonate back to the lungs for CO2 removal. The plasma concentration is about 100 milliequivalents. The intracellular fluid is low since proteins and bicarbonate are abundant in the cytosol and are both negatively charged. They also are always present there, so there's this electrostatic repulsion keeping the chlorides repelled. We do get chloride along with sodium in the salt we eat, and carrier proteins in the renal tubules allow for the reabsorption of sodium and chloride in the nephron loop. The daily loss is relatively small and variable. Hydrogen ion are probably, from chapter to chapter, pardon me, from chapter to chapter, the balance of our hydrogen ion and our acid-base balance is one of our more closely watched chemical conditions, because hydrogen ions, protons are simply so reactive. And so we have acid in our food, and we have a water and acid to acid effect every time we make a CO2 molecule. So we see different acids and bases with different characteristics in our body, and yet somehow the plasma of the blood is balanced in a narrow range, 7.35 to 7.45, a tenth of a pH unit is what our body can maintain, with 7.4 being sort of the median set point. Strong acids and strong bases generally are called that because they dissociate completely. Weak acids have an intermediate stage like our bicarbonate ions. Some molecules remain intact and liberate fewer hydrogen ions, producing a buffering effect. That means that the bicarbonate is there. If we add acid by our diet, it basically converts some of that bicarbonate into carbonic acid and consumes the excess, so the pH is moderated. Fixed acids are those that result from specific reactions. They notice here the breakdown of amino acids, nucleic acids, and phospholipids. Now they stay in solution until the kidneys eliminate them in the urine. Organic acids from our metabolism, producing in that ATP producing pathway, they do not build up unless we go anaerobic. That will build up and require a long period to remove. Volatile acids from CO2, carbonic acid, leave solution and can exit through the lungs, as we've seen. We gain hydrogen ion through our food and also through cellular metabolic activities, anything that produces CO2. We eliminate at the kidneys by putting excess into the urine. We can also eliminate at the lungs by exhaling CO2. If we don't keep this in balance, then it produces an acidosis reaction. Pains and tissue damage do result. I'm going to skip some of these that we have explained. The illustrations are there for your kind of the validation of the flow. The hemoglobin buffer system is one that is active. Hemoglobin itself can act to moderate hydrogen ion changes. CO2, which is the main active molecule, enters the red blood cell itself. By no transport mechanism is required, as carbonic acid is produced by the enzyme carbonic anhydrase. The dissociation to bicarbonate ions produces an increase within the cell. It diffuses out. It's exchanged for chloride ion. This is called the chloride shift. And hydrogen ion are buffered by being absorbed by the hemoglobin molecules themselves. This is shown, I think we saw this CO2 pickup as part of our respiratory system analysis, CO2 being shown here as binding in significant quantity to the hemoglobin molecule itself. At the lungs, because we are enriching this space with CO2, but we're exhaling it, it's leaving the lungs, that produces a lower concentration here, and this CO2 is constantly diffusing in this direction. As the CO2 leaves, it causes the hemoglobin to release, it causes the bicarbonate and carbonate entities to reverse. So here's hemoglobin releasing hydrogen ion to produce carbonic acid and dissociate into water and CO2. This is produced by the activity here in the lungs, a typical source-sink relationship. The sink for CO2 is the lung space. The next time you breathe out, that CO2 leaves. That means there's little CO2 here, and the CO2 that's abundant in the bloodstream diffuses across. Now, as you clean the CO2 out of this blood cell, the blood pressure pushes that out of the capillary and replaces it with a red blood cell that is loaded, and the process of diffusion continues. So these are basically, those slides basically summarize what the printed text has just presented. So we do see acid-base problems. Respiratory acidosis is a challenge to the pH range when the respiratory system can't get rid of CO2 in sufficient quantities. Hypercapnia, low plasma pH, and the primary cause, hypoventilation, breathing too slow. Hyperventilation can, in fact, in some cases, moderate this condition. Metabolic acidosis results from a couple of causes. If you go anaerobic and produce lactic acid in fight or flight situations, that lactic acid is a more permanent chemical substance. In fact, if you go in extreme exertion and go anaerobic, one of the effects of that the next day from overuse of your muscles or overextension of your physical capabilities is a sense of swelling and soreness in your muscles. That's due to the lactic acid that is going to be slowly metabolized and removed. It's going to take several days for that soreness to wear off. In some cases, the production of this produces ketone bodies, which is another effect of the post-absorptive state. It's a problem during starvation or diabetes, and it produces an acid condition. The second major cause, if hydrogen ion cannot be excreted by the kidneys, then the pH can fall in the body. Finally, if you lose lots of bicarbonate, it leaves behind hydrogen ion with nothing to react with, and metabolic acidosis. So that's the story about fluids in the body and some of the very major ions and the relationships that affect them.